Well, thank you, Russ, for that really excellent presentation and the many insights from your distinguished career. I hope you can hear or imagine anyway 340 some people joining me in applause for your talk. One of the things we miss with these virtual talks, but well, we'll start the Q&A session now. But first I'd like to announce, as I said earlier, that point out that the New Phytologist Trust, which has hosted this event, has now been renamed the New Phytologist Foundation. The trustees took this decision because we thought it would better communicate the organization's mission and ethos to the broadest range of the plant science community. The foundation is a nonprofit organization that owns and produces the journals New Phytologist and Plants, People, Planet. And then all of the excess revenues from these journals are reinvested into the plant science community in the form of original support for original symposia and workshops, grants, sponsorship of conferences and events, awards for early career researchers like the New Phytologist Trust, and of course, our support of ESA through this plenary lecture series and awards for early career researchers. But with that, we'll move into the Q&A, and please type your questions into the Q&A box, and if you see other questions that are of particular interest, that you can click on the thumb and it'll move it up, and I will take more notice of it as I try to read the questions to Russ. What I've seen so far, many of the questions are in the sort of ecological or management realm. Let me start with one of the ones posted by, let's see, they're bouncing around as people put new ones in. From Jenny Cribbs, who asked a question about the poplar plantation work you did. Are isoprenes equally important for seedlings and saplings as with mature trees? And how long did the poplar plantation study follow the trees? And then sort of related and in a more general sense, Keith Klein asked, where did that go? Approximately what share of isoprene emissions are attributable to managed perennial landscapes, such as poplar plantations or willow, or introduced plants such as kudzu that you mentioned, versus more natural unmanaged landscapes?
Oh, great questions. Can everybody hear me? Is the sound okay, Rich?
Yeah.
Okay.
Yeah.
In terms of the first question, isoprene emissions are probably quite critical to survival, especially in stressful environments in seedlings and saplings. There is an interesting leaf age effect. That is, when leaves are quite young, isoprene emissions tend to be quite low. And this conflicts a little bit with the need for young leaves to protect themselves against herbivory, especially. And so that's an issue that needs to be resolved in terms of the role of isoprene in potentially mitigating growth defense allocation. But mature leaves, even in young saplings and seedlings, would emit large amounts of isoprene, and presumably the isoprene has a role in protecting those plants from abiotic stresses. And of course, given their proximity to the ground and the microenvironmental effects that may amplify some of those stresses, thermal stresses in particular, isoprene could be quite important in protecting those plants. I think there was a question about the plantation. We actually established two plantations, one in Oregon, which was tracked for 2 years, and one in Arizona, which was tracked for 4 years. The trees were coppiced each year in both of those plantations at the end of the season in order to estimate biomass production. So the shoot growth each year was new growth. I think that may answer one of the questions. And then the question on isoprene emissions for managed and unmanaged landscapes. I don't have a quantitative breakdown of that. It's shifting continually because of the expansion of managed landscapes globally. Isoprene is emitted in high amounts from eucalyptus, from palm species, acacia species, a lot of species involved in managed forests. It's not emitted in high amounts from pine species, so many of the temperate plantations in the southeastern US, for example, with pines, monoterpenes would be emitted in higher amounts than isoprene from the conifers. The conifers are an interesting case study. There's not a lot of isoprene emitted from coniferous species. Tend to rely more on monoterpenes. There are a few, some spruces have been detected as fairly high isoprene emitters, but generally pines and some of the more common species in managed landscapes don't emit high isoprene amounts. So I don't have a quantitative amount of percentage of what fraction of global emissions is due to managed or unmanaged landscapes, but it's significant in both cases.
So Russ, Susan Day asks if you could reflect on the implications of your isoprene results on discussions of BVOCs in cities, especially in the face of climate change.
Yeah, so this has been an issue that's been discussed for a few decades now. When I first got into this line of research in the early 1990s, we were funded by an EPA funded project called the Southern Oxidant Studies. It was actually a joint public-privately funded venture. The automotive industry of America also supported those studies because they were interested in figuring out if these trees were contributing to ozone pollution in urban landscapes more than automobiles. Wanted to get the monkey off their back, if you will. And so, especially in regions of the southeastern US, places like Memphis, Knoxville, Atlanta, these are basically urban forests. If you ever fly over these urban regions, you'll see buildings popping up through the canopy of trees. And most of those trees are oak trees, which the North American oaks tend to be high emitters of isoprene. And so you get a lot of biogenic volatile organic carbon going into the atmosphere, as well as the hydrocarbons from tailpipes. And those biogenics tend to be much more reactive in those regional atmospheres and contribute to ozone more than the tailpipe emissions of isoprene. Now, the problem is that there's a third component that's required for isoprene or any hydrocarbon to generate ozone, and that's nitric oxide, NO, NOx compounds that are emitted from tailpipes. Some of you that are as old as I am might remember Ronald Reagan in 1980 making a comment that on the campaign trail, something like, well, you know, vegetation pollutes more than automobiles, so let's not go overboard in regulating automobile emissions. And he got a lot of flack for that. There's a sliver of truth in what he said, in that the hydrocarbons from trees, in fact, do contribute to ozone in these urban areas, especially on some of these really hot heat episodes that occur during the summer in these urban areas. But what he left out was that the NOx emissions from tailpipes are just as important as the hydrocarbons, and of course those are predominantly in those areas from tailpipes, not from soil NOx emissions, though that does contribute.
So I think there's several questions related to species differences and some of the phylogenetic aspects you talked about early on. Jeff Dukes asks, within species, how much variation is there across natural populations in the tendency to emit isoprene? Has any population level variation been associated with differences in environmental variables or values of other traits within populations?
Yeah, thanks, Jeff. Great question. That's an understudied topic, really. You know, technologically, it's been difficult to make these measurements in the field for some time. And so to get broad surveys within a species has been less emphasized than trying to look among species. But having said that, there have been some interesting studies that Michael Stout has conducted in France in using monoterpene emitting oak species. So there's an interesting phylogenetic story concerning the oaks and terpene emissions. The North American clade of oaks tend to be isoprene emitters. The European clade of oaks, and these two clades split some time ago, the European clade emits monoterpenes, and it's sort of rare to find a broadleaved species that emits monoterpenes at nearly the same rate as isoprene, and this is unusual and unique to oaks. But the European oaks are monoterpene emitters, and they have a monoterpene synthase that's analogous to the isoprene synthase in the North American oaks. And Michael did some studies on variation in the monoterpene emissions from the European oaks within populations in France, and did find some significant variability. And it looked at first cut like that variation sorted itself out as being a little bit more, if I'm remembering right, and it's been some time, I think this is from the late 90s, sorted out with being a little bit more significant in the drier areas that he surveyed. So there might be a little bit of anecdotal evidence there that's somewhat analogous to what we could apply to the case of isoprene emissions. But again, this is really understudied. There's some clever young researchers in this area. I think I discussed some of the studies by Ty Taylor, who has been working in tropical areas, and he's developed a field portable instrument that's unique for moving around in canopies, and you could also use it to move around within a population and survey some of the isoprene emitting capacity. So I think those studies are coming, but they're not there yet.
So along the same line, now I think you probably already touched on some of this, but expand on it a little bit. Is there a relationship between isoprene emission and different categories of plants, such as AM versus EM, early, late successional, gymnosperm versus angiosperm?
Yeah, mycorrhizal status. I don't think I don't think we can nail that one yet. You know, okay, so I think I'll just leave that there because I really don't have something to say on that. Angiosperm, gymnosperm, like I said, it's mostly present in angiosperm species, trees. It's mostly present in woody species. I think I brought that out in the lecture. The strange thing is, though, that we find it deep into some of these phylogenetic relationships, and that we can find it in mosses, we can find it in ferns, and so there's a lot of work to be done on figuring out if there are any ancestral connections there, or if these really are just all independent events. I know Tom Sharkey's been studying the terpene synthases in some of these groups and trying to figure out that question. Just based, like I said in the lecture, when we look at the phylogeny, we can see this thing moving in and out of clades fairly easily, and it is a single gene trait. So there's quite a bit of variation, and that's an interesting story in and of itself. And again, that's been an understudied story. Most of the research on isoprene emissions has been either from people interested in the atmospheric impacts, and so you really look at isoprene emissions from plants as being an input to their atmospheric models. So there's that interest. And then those of us at the very biochemical, physiological sort of end of things, myself, Yogi Schnitzler in Munich, and Tom Sharkey have been doing some of the work, as well as a bunch of other people, Nick Hewitt in England, and we've mostly attacked it from the physiological biochemical side. So some of these issues like phylogeny and like Jeff's question on population level variability just really need to be studied. And so, you know, it's out there for people to start looking at this. And again, some of the young people coming into the field are really beginning to take this up and look at isoprene within the context of structuring communities. Does its effect on abiotic stress tolerance affect community structure? And of course, population level variability would be another one.
So there's several more questions related to phylogenetic aspects, and I think you've already touched on a lot of it. So maybe you can later type in some answers to those questions, and maybe move into more of the physiological arena and biochemical. A question, real simple question, is the amount of isoprene emission different between sun leaves and shade leaves, between upper canopy or upper canopy leaves and understory plant leaves?
Yeah, short answer is yes. Light acclimation, the growth light regime has a significant effect on the isoprene emission capacity. So isoprene emissions varies within a single leaf. If you put a leaf in a gas exchange chamber and you vary the light, you vary the temperature, you vary the CO2, you'll find isoprene responses tracking changes in each of those, not always in the same direction as you would expect. One thing we know, and I touched on this a few times in the lecture, is that isoprene is tightly coupled to photosynthetic capacity. Some of the early studies that Ray Fall and I did in the late 90s showed fairly clear, almost linear correlations between photosynthetic capacity and isoprene emissions. And this is a chloroplast based process that produces isoprene. So you can imagine even ontogenetic sort of controls over scaling, like chloroplast number in leaves, chloroplast density, things like that, will all affect photosynthesis, photosynthetic capacity, I should say, and isoprene emission in the same direction and highly correlated. So yeah, shade leaves deeper in the canopy with lower photosynthetic capacities tend to have lower isoprene emission rates, and sun leaves higher in the canopy with higher photosynthetic capacity, higher isoprene emission rates.
Would you follow up on that? You mentioned just in your acknowledgement slide, I think, of the work you've done with elevated CO2, and of course that's something I've been interested in. Can you summarize a little bit of what you learned on the elevated CO2 interactions?
Yeah, the CO2 effect has been one of the more intriguing effects that we've been working on for some time. So let me start by saying, if you look at the light response of isoprene emissions, it's a rectangular hyperbola, just like photosynthesis. It tracks photosynthesis almost in lockstep. And that's probably due to the requirement for photosynthetic reductant in the MEP pathway that produces isoprene. But when you increase CO2, the photosynthetic rate goes up again, sort of in this rectangular hyperbola that we're all familiar with, described by really well-known models, for example. But isoprene emission rate goes down as CO2 concentration increases. And this was puzzling for a long time. We've published a few things hypothesizing that it has to do with the second substrate required of isoprene emission, and that is pyruvate, and that pyruvate comes predominantly into the chloroplast in the form of phosphoenolpyruvate, where pyruvate kinase converts it to pyruvate. And we were conjecturing that at elevated CO2, the transport of pyruvate into the chloroplast was limiting, and it caused isoprene emission rate to go down. So that uncouples it from photosynthetic rate and the production of photosynthetic products that go into isoprene emission. So that's an interesting phenomenon, and it's caused a lot of us to conjecture on what might happen in the future as CO2 concentrations increase. It looks like most projections are that there's this duality that we need to figure out. As temperature goes up, isoprene emissions should go up. As CO2 goes up, isoprene emissions should go down. So how those two balance themselves out has been a bit of a challenge. And there's a few global models out there that make projections about this, and you might look at that if you're interested in the debate that's going on with that.
So related to the physiology of photosynthesis, Norman Cohn asks, Why do you think that isoprene synthesis did not increase photosynthesis rate? Since isoprene increases chloroplast synthase and protects the photosynthetic apparatus, I really expected isoprene emitting poplars in Arizona to have higher assimilation rates.
We all did. I mean, the prevailing theory when we got into that line of research was that that isoprene has a role in protecting the photosynthetic apparatus against a number of stresses. And so certainly in a place like Arizona, and even though these trees started out being well watered, we also tested them under water stress. We didn't find an effect on the production. In the paper, and I'll refer you to the paper to get a better discussion of this, we're suggesting that the proteomic analysis showed us that while some critical processes to photosynthetic protection were decreased in their expression when we took the isoprene out of the trees, other processes increased in their expression. Alpha tocopherol, for instance. Alpha tocopherol seemed to be suppressed in its expression in the non-isoprene emitting lines, but it seemed to be elevated. Did I get that right? No, it tends to be suppressed in the isoprene emitting lines, but elevated in the non-isoprene emitting lines. And so the thinking being, and there were some others we found as well, the thinking being that when you take isoprene out of the plants, some other processes are increased in their expression that could potentially compensate, especially in the case of mitigating free radical generation during photosynthesis or something like that, in the absence of isoprene. And so that may have leveled things out for the emitting and non-emitting lines. That's one hypothesis.
Mm-hmm. Well, I think related to that, Steve Matzner asked, back to the phylogenetic aspects, I guess, but you mentioned that many species have lost the ability to emit isoprene. Are there any patterns with those species that lost it, such as being less woody? And I've sort of, also when you presented the model of the defense trade-offs, you talked about gaining isoprene and how that shift, but what about those lineages that lost it?
No, there's no evidence that, well, I guess we haven't examined yet, and that's why there's no evidence, whether the plants whether plants that have reverted from woody habit to more woody habit to less woody habit have reversed their isoprene emissions. That's a good question. That's a great question. I don't have any insight into that at the moment. In terms of your elaboration on that, Rich, the phylogenies that we've looked at most, that I've looked at most intensively, particularly those in the Fabaceae that we mentioned. Of course, a lot of those species are African or South American. They're tropical, subtropical in their distributions, and we find in that family isoprene emissions being just as abundant in dry tropic species as we do in wet tropic species. I don't think we can make a distinction there. Being tropical and near and subtropical, of course, all of those trees would have fairly high needs for defense. I mean, we find biotic interactions sort of by conventional theory increasing as you go to the lower latitudes, and so that would suggest that isoprene has a critical role in sustaining those defense requirements, especially in those tropical, subtropical trees. And so we haven't really been able to find something that goes across a defense gradient. That'd be the interesting thing I'd like to look at, is when you go across a defense gradient, do you find these emissions coming and going in a way that tracks that or makes sense? And no, that hasn't been done yet.
Well, there's several questions about the modeling approach you presented, I think, on that defense trade-off. Beth Pringles says, I really like the potential of the model presented at the end to put isoprene at the center of plant trade-offs. But if isoprene emitting can help bridge that trade-off, then why is it so labile? I.e., why do plants lose isoprene emission? Well, you were just talking about this, as often as they seem to gain it, if I understand the phylogenetic patterns. And you were just addressing that, I guess.
I did, but let me just add to how I just addressed that. to narrow this a little bit more, in that the trade-off interaction that I'm describing in that model involves growth, defense, and abiotic stress tolerance. And so the idea that might come into play here is that in species, in clades, in groups that migrate or find themselves in less stressful conditions in terms of abiotic stress, we might find that they are able to handle the growth defense trade-off requirements without isoprene emissions. I don't know. Now, isoprene does come at a carbon cost. It comes at an energy cost. I mean, it's an alkene molecule that's energetically expensive to synthesize in terms of reductant, and it also has a significant, though small, carbon cost. In an unstressed tree, an unstressed plant, isoprene emissions will be about 1 to 2% of photosynthesis rate. Globally, 1 to 2% of GPP. But as you increase stress in the plant, say under a prolonged drought, photosynthesis rate goes down. It turns out isoprene emission rate doesn't track photosynthesis in chronic drought conditions. In fact, isoprene emissions are much more robust as the drought progresses. So isoprene emissions could be very important in sustaining that growth defense trade-off requirement under chronic drought conditions or frequent intermittent drought conditions. And that could be relaxed as these species move into a more moderate environment. But again, this is all, I mean, a lot of let me back up and just provide a little context. For about 20 to 25 years, we've primarily been debating isoprene's role in plants as being a single molecule protecting the plant from high thermal stress or chronic drought stress or ozone stress. And so this whole interaction with the growth defense trade-off and its potential role in that has just emerged in the last 2 years or so as we've gotten access to these proteomic and transcriptomic studies. This builds well into the theme of this year's ESA conferences. Big data sets, when we started looking at these and having access to being able to manipulate this trait and pass it into Arabidopsis or to remove it from poplar, two species with really well-known genomic records, we've been able to see its broader role. So all of this is just really emerging recently, and we haven't had time to put it while I'm hypothesizing this scheme that I presented in the presentation. And we'll elaborate on that. There's actually a paper that Tom Sharkey and I and Yogi Schnitzler are working on at the moment that's almost ready to submit. And so this will be coming out, and I'm hoping it generates a lot of interest in some of these questions that people are having in this growth defense arena. But I'm sorry I can't say too much more deeply on some of these issues at the moment.
That probably includes then no data to answer Ned Fetcher's question about whether you see differences in levels of herbivory or fungal damage between isoprene expressing and unexpressing plants.
No, Ned, not directly. Again, great question, Ned. What I will harken back to, though, is this correlation that we find isoprene emissions, at least at first glance, much more common in tropical, subtropical species. The diversification appears to be greatest at the lower latitudes. And so, following an assumption of latitudinal gradients in herbivory and in fungal pathogenesis, we might hypothesize that that isoprene emissions should track that down into tropical, subtropical systems and diversify much more in those systems than in temperate systems. So we can make the prediction based on theory at this moment. And right now, the first glance analysis would suggest that isoprene emissions being most abundant and diversified in tropical systems follow that hypothesis, support the hypothesis. But let's test it a lot more with within a phylogenetic framework before we really start start going there and being able to assign certain causes to some of these patterns.
Okay, ethylene. Skip Van Bloom asked if there's connections between isoprene and ethylene, since both gases help maintain plant function during stress. Ethylene with wounds, salt, flood, wind, etc.
Right now, no evidence of that, and we have looked in some of the transcriptomic data, and there's no evidence that isoprene is involved in any of the transcription factors involved in ethylene signaling or ethylene production itself. One reason that we're beginning to move toward proposing a role for isoprene in the growth defense trade-off is its apparent dominant role, not dominant, apparent important role in adjusting jasmonic acid pathway signaling and potentially gibberellin signaling as well. So you know, jasmonic acid signaling is really involved in defense signaling, and gibberellin signaling is really involved in growth signaling. So there's sort of the growth defense underpinnings right there in those two pathways. And it seems that isoprene does have very active roles in modulating transcription in those two pathways. And those two pathways have a lot of crosstalk. You know, there's been a lot published in recent years on shade tolerance in plants and its effect on growth, on growth and defense, the growth defense trade-off. And there's some phytochrome involvement in that. So we know that those two, that the growth and defense pathways interact with one another through this crosstalk, and we often find situations where when growth is amplified through gibberellin signaling, for instance, jasmonic acid signaling is muted. There's another pathway that's really important in this is salicylic acid signaling, which is very important for fungal infections, and some of the questions have brought this up as well. And right now, there's no, beyond the fact that the salicylic acid is a product of the shikimate pathway and the phenylpropanoid pathway, both of which are highly upregulated in the presence of isoprene. We should expect that salicylic acid itself should be upregulated as well. But the results are mixed on that. And so I don't think we can yet say anything definitive about the role of isoprene in salicylic acid signaling. Ethylene signaling, I know some recent studies that Tom did at Michigan State, Sharkey, that there was no effect on ethylene in those studies. That was with Arabidopsis that had been transformed to emit isoprene. No studies yet on isoprene ethylene interactions in native emitting species like poplar or oak or anything like that.
So I see two questions here that sort of branch off from what you were just saying. They're different sorts of questions, but perhaps you can try to address them together. John Drake asked, how can mathematical models of these complex pathways help us to test hypotheses regarding the role of isoprene, particularly given the highly interactive and context-specific role of isoprene? But then in a more specific question, Ty Taylor says, what's the relative timing of production and lifetime of compounds like zeaxanthin compared to isoprene? If isoprene is regulated over minutes, I've wondered about how much it is coordinated with regulation of compounds produced more slowly and with much longer residence times in the cell. So, and you know, point out we just have 4 minutes left in the session, so.
Okay, remind me of John Drake's question.
He's wondering about how mathematical models,
right, okay.
So how complex the pathways are, how they can help.
Right. Let me hit that one and then I'll hit Ty's question. Yeah, the modeling to this point, we have pretty good models of isoprene emission in terms of its instantaneous responses to light, temperature, CO2, and those models are based on isoprene's production from dimethyl allyl diphosphate, which is a product of this MEP pathway, the chloroplast pathway. So that one pathway, we can model isoprene emissions. But now with this new broader perspective where it seems like there could be feedbacks and stimulations and interactions, complex interactions beyond that single chloroplast pathway, I still think we can probably do a pretty good job at modeling isoprene in terms of its short-term responses pretty well with the existing models. But what we're hoping, and what we've never been able to do, is model isoprene emissions on the basis of its basal emission rate, that is, its genetically controlled emission rate. And this is where this new perspective may provide some support. We may be able to get a better bead on how to actually predict which species produce isoprene. Right now we can't. In all of the global models, land surface models that Alice Bintler's produced and people like that, we pretty much have to prescribe which species produce isoprene in which landscapes. Maybe we'll get beyond that, but it will be a complex modeling exercise. Ty's question, in terms of zeaxanthin, well, you know, this zeaxanthin is part of the flexible carotenoid cycle, and so it's hard to just talk about zeaxanthin and isoprene in terms of timescales, because there's this violazanthin zeaxanthin sort of exchange, that interconversion that goes on in response to the need for photoprotection. But one thing I would say, Ty, is that I think our perspectives on how long-lived some of these species, these chemical metabolites are, and what their turnover rates are, may change in the coming near months to year sort of time scale. And I have a hunch that while we might not find production rates as fast as isoprene, which tends to fluctuate on the order of seconds or less, that we will begin closing the time frames on, so we will find within day. In fact, a lot of the metabolites we found expressed through the proteomic analysis varied within a day. And so I think we're going to find a lot of these expressions on much shorter timescales than we originally thought, and we'll begin converging on the metabolic metabolite perspective and in a broader sense, and the isoprene perspective in a more narrow sense.
Well, I think we've reached the end of the time. There are remaining questions, and sorry I didn't get to them all. I don't know if, Russ, you had the opportunity to give any written responses to them in the chat box. There's one question, maybe you could give completely different from others, maybe you can give a 10-second answer to. Could isoprene emissions be detected using remote sensing, similar to what Rob Jackson was just describing for methane?
Not isoprene directly. Its emission its absorption spectrum is not one that we would be able to detect at present. But the products of isoprene oxidation in the atmosphere are actually quite detectable by satellite, and there's been some studies on that. And so I'll let you go to Web of Science and look those up. But yes, there are some remote sensing analyses of isoprene emission globally that are dependent on satellite platforms.
Well, thank you very much, Russ, for your talk and for engaging with the community here during the Q&A session. And thanks for everyone who's been listening in and asking really great questions. So
thank you. See y'all.
Okay, bye-bye.